Hello and welcome to another lecture of the course Aquaculture. Within the major topic of age and growth estimation and its significance in aquaculture, today we are going to talk about fish age estimation. If you can recall my previous lecture, I told you about the significance of age and growth studies, why they are important. Now, when we understand why, then now the time is to understand how. So, how fish age can be estimated? There are different methods of studying fish age. We all know that fishes are not going to tell their age themselves, so we need to have some kind of provisions to get robust information, the precise information on their fish age, their age. So there are methods like direct method, length frequency distribution method, annuli or hard anatomical structures, annuli on hard anatomical structures method. We will discuss one by one. Direct method, there may be direct observation of fish in confinement, or we can mark them or tag them and then recapture them. So these two major techniques. In fact, direct observation of fish in confinement is nothing but keeping the fish population in confinement where you can monitor their entire lifespan, and you can study that what are different age related changes occurring in the entire life cycle of the fish. So whenever you need to have information on age and you wish to correlate it with any kind of biological or physiological characteristics, then you have the age information, and that is the most correct, authentic information that we can have. But this is not possible everywhere. This you can do just for the academic or research purpose. But if you need information on the age for some other reason, or you are managing natural water body, this is only possible in confinement. If you are doing it in natural water body, then there are many other factors which are operating, and it is not possible to go for this kind of information on age. And practically, we are having majorly this kind of information where we cannot get enough input on age of the fishes from the natural environment. So that is one of the limitations, that we cannot use this. Then we have marking, tagging and recapture technique. This is very simple technique, and this has been used since beginning of studying this kind of age related biological characteristics in fish. What we do here, we use certain kind of chemicals that can leave mark on the certain structures of the fish, and whenever we recapture those fishes, we will have the information that okay, on such date the fishes were marked with this kind of mark that you can reread, and at that point of time they were having these biological characteristics. So whenever you recapture, you can correlate that, and you can continue with your studies or whatever information you are looking for. It's not only that chemicals are only being used for marking. There are other methods also. For example, thermal marking is also there, but usually chemicals are used. And in fact, among the chemicals also, there may be different chemicals that can be used, but tetracycline is very popular, and that has its own advantages. And when we use this chemical, then what happens that as you can see here, that you will find fluorescent marks on the calcified structures that can be reread using UV light whenever you recapture the fish and you wish to get more information on its age. So marking is one of the method, as in the previous slide you have seen, and then tagging is another one method that is also used. Marking and tagging has been used for quite long time to have fish biology studies in many countries. Tagging, what is tagging? You put some kind of tag, some kind of coded information written on the tags, and you tag the fish with that material. For example, here some tag is being attached to the fish. There are different kind of tags you can tag in the say, for example, in the jaw, you can tag in the fins, you can tag in the tail, you can tag somewhere in the muscle. So there are different methods, different techniques of tagging that you can go through in some separate lecture. And what happens that these tags may be of some material which is inert and which is not interfering with any kind of biological activity of the fish, and you will have to write some kind of coded information on the tags. So whenever you recapture, you just see the code, decode that information, and you will have everything with you, that at the time of tagging what could have been the age of the fish or whatever other information you have written on that. So that is one of the methods, but these are time consuming and they require little expertise, so they are not so popular studies. Length frequency distribution method. Uh, Length frequency distribution method, this is based on the concept that here what we do, we in routine we sample large number of fishes of almost all size groups and then count their number, believing that we have collected the samples which is representative of every life stage of the fish present in that particular area. So whatever candidate species population you are studying, you believe that you have collected the data related to length, weight and number of all representative size groups, and on that belief we then plot the length and frequency and length versus frequency, and then we get some kind of peaks, and that peak, every peak is supposed to represent one age group. So that is the basic belief that what we do. Here you can see that it is written, length frequency data are used in various analytical, graphical and software assisted techniques to estimate age, growth and other population parameters. Simple, fast, but rather inaccurate method. I will explain you why. It can be used with fish species which breed only once in a year. This method can be very good for young fish, but becomes problematic when you go for older fishes and your population may be having all the groups. What happens? I told you that this is on the belief that how peaks are formed. As you see here, the sharpness of the peak decreases with increasing age, and if you extrapolate these lines like this, like this, like this, then you will see that there is increasing overlapping when you go to increasing age. Therefore, this is often considered that the information from length frequency method may be useful for only younger fishes where the sharpness of the peak is very good, but not for the older fishes. For example, in age 3 you can see here the peak is almost not identifiable. And while going for length frequency method, we have to take certain precautions. For example, we should try to do fish sampling from different regions of that water body, whatever we are selecting. We should try to take a sample on the same day if possible. We should try to collect the samples representative of all size groups. So, for example, 0 to 10 centimeter size, then 11 to 20 centimeter size, then 21 to 30 centimeter size, like that. So, you will have to get information on, data on all the representative groups, And again, the fish should be breeding only once in a year. So if these conditions are met, then we can go for length frequency method. Even then, the data is not as reliable as the direct method or other methods of hard anatomical structures. But when we are dealing with large water bodies in oceans, then this information also becomes very crucial and often helps us to get very good management decisions. Now, the most popular of the age estimation methods is determining fish age using hard anatomical structures. In fact, this is on the belief, on the concept that fishes grow throughout their life. Fishes show growth throughout their life, although the pace of growth, the rate of growth may slow down. And when fishes grow throughout their life, that means the different body parts of the fish also show growth rate throughout fish life. There will be proportional growth of any structure, any body part to the total body growth. Based on that concept, the structures like hard structures like bones, any bone, spines, otoliths, scales, etc., they may also show proportional growth to that of total fish growth. And when fishes are growing, we know that there are certain times when fish shows very good growth, when food is abundantly available, when environmental conditions are very conducive, then fish will grow at a very faster pace. That point, the bony structures or calcified structures will show light zone on them. And during adverse conditions, for example, during harsh winter conditions or during breeding season, when many of the fishes almost stop feeding in these regions, when fish growth becomes slow, then the calcified structures start showing dark line, dark zone. So, dark zone will represent slow growth, light zone will represent fast growth, and this is reflected on the calcified structures or bony structures found in the fish. So, that is the basic concept, that we have this kind of information on the calcified or bony structures. We have to interpret this and get to know that how many dark zones are there or light zones are there, and accordingly we can say that what is the age of the fish. Here it is written, hard parts grow by deposition of different minerals in a biological matrix. Any changes in growth rates may be reflected as zones or bands. By tracking down these inscriptions, age of the fish can be determined. All this I have just explained to you. During slow growth phase, rings or bands will be laid close together, and whereas during fast growth phase they will be laid far apart. That's why lighter zone appears, and when they are the growth lines are very close to each other, then we have dark bands or dark lines or dark zones. Among hard anatomical structures, there are many which are used for popularly for fish age estimation. For example, scales, opercular bones, otoliths, spines, fin rays, vertebrae, etc. You can have, in fact, you can use any calcified or bony structure if you are able to process and prepare that aging structure well, where these growth rings are clearly seen. If you can process that, you can read that. We will discuss one by one these important structures. Scales. Scales, many of you may be knowing that scales are probably one of the most popular methods for fish age estimation. In whichever fish, if we see scales, then we go for scale collection and prepare it, process it, and read the age of the fish. So, this is very popular because this method is non-lethal. Plucking few scales from the fish body will not harm it. So, you can pluck it, you get the information on age, and then fish will be fine and you are also having the information. But the problem with this method is that since we know that this is the outermost part of the body of the fish, so it will be subjected to wear and tear of the environmental conditions. You will see that the fishes may also rub their body against some hard substratum, or in the regions like caudal peduncle, which is very active during swimming, there may be shedding of scales. So, that is again one of the problems. So, you may not get the scale sample what you are intending to get. And there is one more problem, that when I told you that fish grows very fast during early life and then growth pace or growth rate slows down. When the growth rate slows down, that means the annuli or circuli, in case of scales what we call annuli, which is showing annual pattern of dark lines we usually interpret as annuli. Annuli will be laid down in older fishes at very closely to each other, and when they will be laid down very closely to each other, that means it will be very difficult for us to identify the one ring from the other, and ultimately that may create confusion and we may end up counting less number of age than what fish really is. So, scales may be very good if, for age estimation of younger stages, but for older fishes, scales may be avoided if you are having very old fish. This is the structure what we get in case of scales. As you will proceed towards margin, the gap between two annuli will go on decreasing because of the decreasing growth rate. And when fish becomes too old, this is only 5 years old that has been shown. If fish becomes too old, then these annuli will be placed very close to each other, and sometimes it will feel to you that they are over each other, and that becomes very difficult, and we often end up underestimating fish age. That fish age will be utilized for formulating management strategies and devising policies. And if you are using wrong information, then the policies or management strategies that you will be devising, that may be flawed, and that may result into fishery collapse also. Then we have spines and fin rays. Fish spines, again they are bony structures. And then fin rays, in case of bony fishes, we are having fin rays also. So, they may also be having the same information. I told you that any bony structure, any calcified structure may be having the information. The only thing is that you should be able to work out the method by which we can process or prepare that aging structure to make rings very clear. Here also, in case of scaleless fishes, in fact, when in case of fishes which are having scales, we are having scales which is non-lethal method. Here, if we do not intend to cause too much harm to the fish, then we can go for either spines or rays. So, if you study the age of the fish using this method, then usually we have to cut sections from the base part of the spines or rays. But one thing which is shown here also and here also, that in case of spines, what happens, the lumen part, the central lumen part, there is central lumen in case of spines, fish spines. This lumen part goes on increasing with progress of age, and ultimately it creates a kind of hole, and this may engulf many of the initial age lines, the growth lines which are representing 1 year or 2 year of age. So in older fishes, if you are taking this method, then you may end up underestimating the age again, because the initial annuli are lost to the lumen. Here few more sections with proper reference have been given, that how we study. In fact, we have to interpret these dark lines and light zones. Opercular bones. Opercular bones, you know, in certain fishes, it all depends on the availability of the structure, the ease of processing, and the age reader, what kind of aging structure the age reader is comfortable to read. In case of opercular bones, what we do, we simply take out the opercular bone. You may cut it using scissors, or you may just twitch it with your fingers and take it out. You have to detach all the tissues from the bone. Maybe you can take help of boiling it in water, or sometimes very dilute solution of KOH you can use. Ultimately, when it is clear, then we can dry it and read the light and dark zones. And the dark lines we often interpret as annuli that can be interpreted in many of the fish species. This has given very good age reading information compared to other methods. This is how opercular bone looks like, or gives information to very clear information. As you can see here, the dark lines that have been pointed out, you can interpret that as the annuli. Then we have vertebrae. Vertebrae, they also bear variation in growth, or they also document variation in growth in terms of light and dark zones. Vertebrae, in fact, in case of sharks and skates, the vertebrae is the most reliable method for age estimation. But even in freshwater teleosts, the bony fishes, we use the vertebrae for age estimation, and in many species, vertebrae is found to be providing the most accurate or most reliable age information. Here we have to collect the vertebrae. Usually we collect from the anterior region, usually 4th to 10th vertebrae we prefer. You take out the vertebrae and clean it properly, thoroughly clean it for whatever attached tissues are there, and then dry it, and then you can read the vertebrae. If necessary, you can cut sections of vertebrae also, as I told you regarding spines. And if you want, you can stain it also. There are many methods to study, and you can have very good information on age from vertebrae as well. Then otoliths. Many of you must have heard about this structure, but if you have not heard, they are also called as ear stones. Why? Because they are present in the internal ear. In the internal ear, on both the sides of the head, they are present in pairs. And one important thing about otoliths is that they're a kind of magical structures. They are acellular, and structure wise, they are crystals of calcium carbonate in protein matrix. So that is a crystalline structure, majorly of calcium carbonate in protein matrix. They are located in internal ear, I have told you, and otoliths grow throughout their life, throughout the fish life. They are metabolically inert. They are metabolically inert. They do not participate in metabolism. And one more important thing, they do not show reabsorption even during starvation. So resorption will not be there. They are metabolically inert, they are not participating in metabolism, they are growing throughout fish life, and they are acellular structures. When these kind of characteristics are found in some structures, so what will happen? Whatever information will be there or recorded on such kind of structures, otoliths, that will remain for the entire life of the fish. Some information which has been recorded early in the beginning, like fry or hatchling stage or fingerling stage, that will be there for entire life of the fish. And it's not only entire life of the fish. In fact, during my stay at Munich, LMU Munich, we studied fossil otoliths of Miocene, and we have been able to interpret the growth lines, the annuli in that otolith, and we also have been able to mark even daily growth rates, and by that process we deciphered the larval duration of the fish from that otolith. So, because of these characteristics, whatever information has been recorded as light and dark zone in terms of dark line on the structure, that will be there for long time. This is from one of my publications. My students, they worked on fish otoliths, and they took out the internal ear in order to show that what are three otoliths which are found in pairs in case of fishes. This was from Channa punctata, the commonly known as soli fish. Here you can see that one otolith is called Sagitta, another is called Asteriscus, and the third one Lepilus. These are three otoliths. They are present in the internal ear on both sides of the head, and Sagitta is in the saccule, Asteriscus is in the legina, Lepilus is in the utricle. These are the otoliths which are being shown here. Now, if you process these otoliths, then what will happen? You can see the lines very clearly, and these light and dark zones you can interpret. The problem is that even with these scales, if you are going to study age of the fish through scales, if the scale is good, clear, not very dirty, you can use your fingertips to wash it, dry it, and against a source of light you can see the lines very clearly. But sometimes that is not so easy. So you will have to prepare the structure or process the structure in order to make it more clear to show its annuli. Only thing that you have to take care that you should avoid putting extra lines while cleaning the structure that may create some kind of confusion in identifying the true lines, growth lines, which are to be interpreted as annulus or annuli. Some otoliths are having such a shape that you can get the light and dark zones very clearly, and you can study the whole otolith. But there are certain otoliths which are having very different shape, where interpreting the light and dark zone becomes very tricky and difficult. In those cases, we can go for sectioning the otoliths, and while sectioning the otolith, you will get, the only thing you have to care is that the section must be cut through the core of the otolith, or any structure you are going to cut sections, and then that can be visualized under microscope, and you can get a very better result. Here some of my publications I have shown you that from my lab only. In fact, age and growth studies have been undertaken in our country for long time. But I saw that when the fish from when we are collecting different aging structures like scales, otoliths or opercular bones or vertebrae from the same individual fish, their readings are not same. Why not same? Because our methods of preparation and processing of these structures may vary. So, some show very clear annuli, some do not show. So, which one to select for getting good information? For example, 5 aging structures you have taken from a fish, they are not showing consensus in terms of age reading. So, which one you are going to follow? To remove that fallacy, to solve that problem, what we did, we carried out comparative study of different methods, and there are certain procedures described which were utilized in order to compare the age from different aging structures. And after calculations, after understanding that what is the magnitude of error that we noted in different kind of methods. So, the method which was showing minimum of error and maximum of consensus between independent age readers, we selected that structure and we recommended that structure for age reading in that particular fish species, that is called as precise age estimation in fish. In fact, this is the first publication from the country on precise age estimation in freshwater fishes of the country, India. So, thank you so much for this lecture. We will discuss some other topic in the coming lecture. Thank you so much.